Welcome dear viewers to the video on guidance for the industry, dissolution testing of immediate release solid oral dosage forms. For making this video, I have referred the US FDA guidance for industry, and in this video the principles for dissolution testing, the regulatory requirements, quality requirements, and the basics for dissolution testing are covered. So the dissolution guidance by the US FDA, it can be used for the formulations like tablets, capsules. Also you can use it for oral suspensions, solutions, and many other immediate release type of formulations. Whenever you want to perform the dissolution testing, that time this guidance will help you to know the requirements and to know the principles. So let's start with the video. I request you to go through the videos available on this channel on the dissolution topic, so that one by one you can understand the topics related to the dissolution. Also I request you to go through this guideline or guidance provided by the FDA. After watching this video, you will be able to learn that guideline in much detail. So the US FDA has provided the guidance for the IR formulations. The purpose of this guidance is for the dissolution testing of immediate release type of dosage forms. The guidance provides recommendations on general dissolution testing, how the dissolution testing is to be performed, setting specifications for the dissolution based on the biopharmaceutics, then statistical methods for the profile comparison, and criteria for granting the biowaivers. Also, this guideline complements the SUPAC IR guidance. Now, you might be knowing about the importance of dissolution testing. Dissolution testing is a routine quality control test. It predicts the in vivo drug performance, and it ensures the batch to batch consistency, batch to batch quality. It shows that the generic product will be safe and effective and equivalent to the reference formulation. If it is the novel formulation or new drug application or new formulation, the dissolution testing predicts the in vivo behavior by the in vitro testing. Also, it provides the insights that the processes are consistent and the product is robust as the dissolution is similar across the batches. It supports new formulation development and it supports the post-approval changes. Many of the times the biowaivers can be taken or granted considering the dissolution testing. Bioequivalence studies and basis for the compendial standards. USP monographs are there, which provides you the requirement of the dissolution testing. Then, as per this guideline, biopharmaceutics classification system is also considered. BCS class 1, 2, 3 and 4 are there based on the solubility and permeability of the drug molecules. Class 1 drug substances are highly soluble, highly permeable, and if there is more than 85% drug release in 15 minutes, it is considered that the bioavailability is not limited by the dissolution, because the dissolution is already very high or very rapid. Then BCS class 2, low soluble, high permeable, dissolution is rate limiting for the absorption or bioavailability, and IVIVC will be possible. For BCS class 3, the solubility is high but the permeability is low. So permeability limited is the absorption, and bioavailability, limited IVIVC is possible. For class 4 drugs, the solubility and bioavailability both are low. So IVIVC is very less possible in this case. We cannot predict in much detail, and major delivery challenges will be there. Then dissolution specification types. So based on that, you need to have the dissolution specifications. Single point specification, 2-point specification, and profile comparison will be there. So single point is for the highly soluble drug substances IR formulation. In other words, you can say for the IR products containing highly soluble APIs like BCS class 1 or 3. Single point dissolution specification can suffice the requirement. 15 minutes you can give, 30 minutes you can give as a Q point. And single point dissolution is QC testing for highly soluble rapidly dissolving drugs. Then 2-point dissolution specification. QC testing for slow or poorly soluble drugs like carbamazepine. So you can give the Q point at 30 minutes and 60 minutes or 45 minutes and 120 minutes depending on to the formulation. So you need to consider the 2 points. Only 2 point will be there. And third is profile comparison. So profile comparison is point to point release profile comparison. For example, if your release is up to 1 hour, you need to have the profile for dissolution like 15 minutes at time points like 15 minutes, 30 minutes, 45 and 60 minutes. So it is required in the SUPAC related changes, and also it is required in the BioWaiver support. Then setting the specifications NDA versus ANDA. The formulation may be new or the formulation may be generic. Sometimes hybrid type of formulations may also be there, or novel type of formulations may be there. So how you will set the dissolution specifications for the NDA and ANDA? For NDA, the dissolution specifications are based on the pivotal clinical bioavailability batches, and for ANDA, it is based on the reference listed drug dissolution and based on the bioequivalence batches. Also, stability data is required to be considered. Then it must comply throughout the shelf life. ICH Q1A provide you the information regarding stability data from 3 batches used for setting the specifications. Then approaches for new chemical entities. So if you are working onto the formulation containing the new chemical entity, that time consider the pH solubility profile of the API, consider its pKa, consider permeability, site of absorption, stability in the GI media at various pH. Lot of data is required to be studied and generated. Then generate the dissolution profile using basket apparatus or pedal apparatus. Typically, basket apparatus is operated at 50 rpm or 100 rpm, and pedal generally at 50 rpm or 75 rpm. Some of the formulations I have seen with the pedal speed of 55 also and 60 also. So general pedal speed is 50 and 75 rpm. Highly soluble drugs, a single point Q point can be given like not less than 80% or 85% in some time like 15 minutes, 30 minutes, 45 minutes or 60 minutes, but generally 60 minutes is not given. Generally 15 minutes or 30 minutes is given. Then poorly soluble drugs, 2-point testing can be given and not less than 50% or not less than 80% at 2 time points like 15 minutes, or you can consider 30 minutes, 45 minutes and 60 minutes, or 120 minutes also can be considered for the drugs which are low soluble. This is regarding the approaches. Now approaches for the generic products. So whenever you are working on to a generic formulation, you need to check whether the monograph is available, whether the US FDA has provided the requirement in Orange Book or Orange Book Biopharmaceutics review, or you can go through the OGD media, and then if it is not available, then in-house method is required to be developed. So USP test is available, then use the USP method, add the profile at 15-minute intervals. Then USP test not available, NDA method public. So as the USFDA provides requirement through its Office of Generic Drugs and the OGD media, then use that method which is used by the NDA. And if USP and NDA method is not in public, then test the multiple media, vary the apparatus, develop the dissolution method which is in-house, verify it and validate it. Then special testing cases. So two-tier dissolution are also used or required sometimes with SGF and SIF with or without enzymes. Then mapping surface response technology or methodology, you can say, to link the manufacturing variables to dissolution and bioavailability can be done. IVIVC is required and more feasible for the low soluble drugs belonging to BCS class 2. Then dissolution profile comparison. So different approaches you can use. Model independent approach and model dependent approach you can use for dissolution profile comparisons. Model independent approach is F1 factor 15 equal to less than 15 is required and F2 should be equal to 50 or above 50. Then you can say that your dissolution profile is similar. Model dependent approaches, you can use the models like linear, Weibull and many others. Compare the parameters within similarity region for providing the dissolution profile comparisons and similarity. Then SUPAC IR, it defines the change levels and required testing. So minor changes, major changes are there. I request you to go through the SUPAC guidelines to understand this topic in detail. I have made some videos on this topic. You can search in the channel playlist and go through those videos. Then in SUPAC IR, define the change levels and required testing. For minor changes, dissolution testing is required. For major changes, depending on to the formulation, depending on to the API, IVIVC availability, you can go through the dissolution and bioequivalence. Also, sometimes some strengths are required to undergo the bioequivalence study, while you can get the waiver for additional strengths. Use the F2 similarity factor and other model independent approaches for the dissolution similarity. Then come to the biowaiver. Biowaiver is granted for the lower strengths and also for the BCS based IR formulations. The biowaivers are granted for the lower strengths if the proportional composition is there throughout the strengths, if the API shows the linear pharmacokinetics, and you have to generate adequate dissolution profiles and should provide the justification that all the strengths meet the dissolution similarity criteria and the B strength has shown the bioequivalence. So based on the pivotal strength bioequivalence, you can get the biowaiver for lower strengths. So I have also made video on the biowaivers, additional strength biowaivers requirement. You can go through those videos. Then appendix A is given, which highlights the apparatus, medium and agitation. Then validation. So what is the requirement of apparatus, medium, agitation and validation? Generally USP1 basket, USP2 paddle is used. Alternatively, you can go by USP3, that is reciprocating cylinder, and USP4, that is flow through cell. You can optimize, you can study different media volumes like 500 ml, 1000 ml, pH 1 in a media pH of pH 1.2, 6.8. You can go up to 8 pH if justified. Then SGF, SIF with and without enzymes can be used, and surfactants can be used for the poorly soluble drugs like BCS class 2 and BCS class 4 drugs. Then agitation and validation. Basket with 50 rpm or 100 rpm, paddle with 50 rpm or 75 rpm. Then validation includes system suitability, deaeration if required or degassing, manual versus automated equivalence is required to be shown. Then analytical method validation is required. Now see the main takeaways from this video. Dissolution testing is a very important and critical QC and regulatory tool, and dissolution depends on the BCS class and the in vivo data. Profile comparisons ensure the product sameness after change like SUPAC change, and dissolution supports the biowares and the post-approval stability. So this is regarding the dissolution testing requirement as per the US FDA. I request you to go through this guideline which I have mentioned above. Now, after watching this video, you will be understanding that guideline in a better way. So, if you have any questions, you can mail me or you can comment. Thank you for watching the video.